Hello everyone, now let us discuss about some of the important drug interactions. In the current session, we will be discussing some of the important pharmacokinetic drug interactions pertinent to absorption, distribution, metabolism and excretion, ADME. Coming to absorption interactions. Let us see the table here. Here we have object drug, precipitant drug and the influence of precipitate drug on the object drug. Object drug is the victim here. Whenever we take object drug and precipitant drug together, the precipitant drug will have a effect on the object drug. Here the object drugs are, the mechanism of action is complexation and absorption. Adsorption. Drugs such as tetracyclines, fluoroquinolines like ciprofloxacin and penicillamine, whenever they are taken along with antacids and also food and mineral supplements such as aluminium, magnesium, iron, zinc, bismuth and calcium ions. Mineral supplements containing aluminium, magnesium, iron, zinc, bismuth and calcium ions, whenever they are taken together, it leads to formation of poorly soluble and unabsorbable complexes with heavy metal ions, which alters the absorption and dissolution of object drug that are tetracyclines and fluoroquinolines. Similarly, cephalexin, sulfamethoxone, trimethorphan, warfarin and thyroxine. Whenever these drugs they are taken along with cholesterolamine, it reduces the absorption due to adsorption and binding. Whenever these drugs are taken along with cholesterolamine, their absorption is reduced due to the adsorption and binding. Next mechanism, alteration of GIPH, gastrointestinal pH. Whenever sulfonamides and aspirin, when they are taken along with antacids, due to the alteration of GIPH, the dissolution and absorption rate of sulfonamides and aspirin are increased. Enhanced dissolution and absorption rate. Next drug is ferrous sulfate. Whenever it is taken along with sodium bicarbonate and calcium carbonate, it leads to decreased dissolution and decreased absorption of ferrous sulfate. Next, ketoconazole, tetracycline and atenolol. Whenever they are taken along with antacids, their dissolution and bioavailability have been decreased. Next mechanism is alteration of gut motility. Whenever aspirin, diazepam, levodopa, lithium carbonate, paracetamol and mexiletine, when they are taken along with metoclopramide, the influence of metoclopramide is rapid gastric emptying and increased rate of absorption. Next example, whenever levodopa, lithium bicarbonate and mexiletine, when they are taken along with anticholinergics such as atropine, this leads to delayed gastric emptying and decreased rate of absorption. Next, coming to alteration of GI microflora. Whenever digoxin is taken along with antibiotics such as erythromycin and tetracycline, it leads to increased bioavailability of digoxin due to destruction of bacterial flora that inactivates digoxin in lower intestine. And whenever oral contraceptives are taken along with antibiotics such as ampicillin, it leads to decreased reabsorption of drugs secreted as conjugates via bile in the intestine. Next, coming to malabsorption syndrome. Whenever vitamin A, B12 and digoxin are taken along with neomycin and colchicins, it leads to inhibition of absorption of vitamin A, B12 and digoxin due to malabsorption or steatorrhea caused by neomycin. Next, coming to distribution interactions. The first one is competitive displacement interactions. Here, the important drug interactions in distribution interactions are displacement interactions. Here, one drug is displaced and other drug acts as a displacer. Let us see the first example. Anticoagulant. This is the displaced drug, and the displacer is phenylbutazone and chloral hydrate and salicylates. Whenever anticoagulants, especially warfarin, it is taken along with phenylbutazone, chloral hydrate and salicylates, it increases the increased clotting time and increases the risk of hemorrhage. Because the warfarin levels are increased because it is displaced from the protein binding site. The next drug is tolbutamide. Whenever tolbutamide is taken along with sulfonamides, it leads to increased hypoglycemic effect of tolbutamide because it is displaced. Next is methotrexate. Whenever methotrexate is taken along with sulfonamides or salicylic acid, it leads to increased methotrexate toxicity. The next is phenytoin. Whenever phenytoin is taken along with valproic acid, due to the displacement of phenytoin, it leads to phenytoin toxicity. Next, coming to metabolism interactions. First one is enzyme induction. Drugs such as corticosteroids, oral contraceptives, coumarins, phenytoin, tolbutamide, tricyclic antidepressants, whenever they are taken along with barbiturates, the plasma concentrations or plasma levels of these drugs are decreased, and this leads to decreased efficacy of the object drugs. Barbiturates, it decreases the plasma levels and overall efficacy is reduced. Whenever barbiturates are taken along with corticosteroids, oral contraceptives, coumarins, phenytoin, tolbutamide and tricyclic antidepressants. The next example is similar action. It has a similar action. Whenever phenytoin is taken along with corticosteroids, oral contraceptives, theophylline and cyclosporin, the plasma levels of corticosteroids are reduced. This leads to decreased efficacy. And similarly, whenever rifampicin is taken along with oral contraceptives, oral hypoglycemics and coumarins, the plasma levels of oral contraceptives, oral hypoglycemics and coumarins is decreased, and this leads to decreased efficacy of these drugs. The next mechanism in metabolism interactions is enzyme inhibition. Previously, it was enzyme induction. Coming to enzyme inhibitions, whenever tyramine-rich foods such as cheese, liver, yeast products, whenever they are taken along with MAO inhibitors, for example, phenelzine, paraglycine, etc., it leads to, due to enzyme inhibition, enhanced absorption of unmetabolized tyramine, increased pressor activity, and potential fatal risk of hypertensive crisis can occur. Whenever the MAO inhibitors are taken along with tyramine-rich foods, it leads to enhanced absorption of unmetabolized tyramine, and this leads to increased pressor activity, and it can cause potentially fatal risk of hypertensive crisis. And whenever drugs that undergo extensive first pass hepatic metabolism, the drugs which undergo extensive first pass hepatic metabolism, for example, propranolol, calcium channel blocker, etc., whenever they are taken along with grape juice, due to enzyme inhibition, it leads to enhanced absorption of drugs and increased risk of toxicity. Next, coming to folic acid and phenytoin. Whenever folic acid is taken along with phenytoin, decreased absorption of folic acid occurs due to inhibition of an enzyme responsible for its absorption. Next, coming to tricyclic antidepressants. Whenever tricyclic antidepressants are taken along with chlorpromazine and haloperidol, this leads to increased plasma half-life of tricyclics and increased risk of sudden death from cardiac disease in such patients. And similarly, whenever metronidazole and phenylbutazone, they are taken along with coumarins, Due to enzyme inhibition, increased anticoagulant activity of coumarins takes place and this results in the risk of hemorrhage. Next, oral hypoglycemic drugs. Whenever oral hypoglycemic drugs are taken along with phenylbutazone, sulfaphenazole and chloramphenicol, hypoglycemia may be precipitated due to enhanced action of oral hypoglycemics. Next, coming to alcohol. Whenever alcohol is taken along with disulfiramide, metronidazole and tinidazole, disulfiramide reactions occur due to increased plasma levels of acetaldehyde. Similarly, when AZT, which is nothing but azidothiamidine or mercaptopurine is another drug. Whenever AZT or mercaptopurine, they are taken along with xanthine oxidase inhibitors, for example allopurinol, it leads to increased toxicity of these antineoplastic agents due to enzyme inhibition. Next example is alcohol, benzodiazepines, warfarin, phenytoin, theophylline and phenobarbital. Whenever they are taken along with cimetidine, it leads to increased blood levels of those object drugs. Whenever cimetidine is taken along with alcohol, benzodiazepine, warfarin, phenytoin, theophylline and phenobarbital, the blood levels of these drugs are increased. Next, coming to excretion interactions. The first mechanism is change in the active tubular secretion. Whenever probenecid is taken along with penicillin, cephalosporin, nalixidic acid, TAS, methotrexate and dapsone, elevated plasma levels of acidic drugs occurs and this leads to increased risk of toxic reactions. Next is cimetidine. Cimetidine is a base. Cimetidine, whenever it is taken along with procainamide and ranitidine, it leads to increased plasma levels of basic object drugs and results in the risk of toxicity. Next is phenylbutazone. Whenever it is taken along with acetohexamide, it leads to increased hypoglycemic effect. Next mechanism is changes in the urine pH. Excretion interactions which occur due to changes in the urine pH. Whenever antacids and thiazides and acetazolamide, whenever they are taken along with amphetamine, tetracycline and quinidine, increased passive reabsorption of basic drugs occurs and this leads to increased risk of toxicity. These are the basic drugs. Whenever they are taken along with antacids, thiazides and acetazolamide, increased passive reabsorption of these basic drugs occurs and this leads to increased risk of toxicity. Next, coming to changes in the renal blood flow. Excretion interactions which occur due to changes in the renal blood flow. Drugs such as NSAIDs, which are nothing but inhibitors of prostaglandin synthesis, which control the renal blood flow partially by vasoconstriction. Whenever NSAIDs are taken along with lithium bicarbonate, this leads to decreased renal clearance of lithium and it increases its risk of toxicity. Thank you for watching. Please subscribe for further videos on pharmacovigilance and other related pharmaceutical sciences.